and other cellular process. Oh, Michael, there you are. Can you hear us? And I guess you could hear me, but if you unmute, let's try if we can hear your voice.
Okay, can you hear me
Yep.
now?
Yes, perfect. How are
Sorry
you?
about that. I'm good. Yeah, sorry. I'm not sure what was happening before. I could hear you, but
Yeah, I figured because you left and then joined again, and now it works. So great. Thank you so much for coming. It has been a little while since we talked, and in the meantime, so many things happened. You published papers that were really interesting, so we're really happy having you here.
cool. Thanks.
Thank
Yeah,
you so
thanks for
much.
having me.
Yeah, let me start by introducing you just for in case people don't know you. But I guess most people do. So let's keep it maybe relatively short and then go into the three recent papers. And if you, of course, want to talk about additionally other papers or topics from your lab, just let me know. I posted them in the chat, and the first one is pinned on top, and I guess we'll just do, like we did it last time. I'll ask some questions and you go and, yeah, talk about your amazing work. So,
Sounds great.
perfect. Professor Michael Levin, he has a dual bachelor degrees in biology and computer science and a PhD from Harvard in genetics. And then he also did his postdoc at Harvard in cell biology. And then he started first his lab in 2000 at the Forces Institute. And now he is at Tufts University and Harvard Institute. And he is also the director at the Allen Discovery Center at Tufts, and the director of Tufts Center for Regenerative and Developmental Biology. And Mike is a world-renowned scientist, currently a distinguished professor, and his groundbreaking studies in bioelectrical signaling in cells and tissues, and also designing and shape complex organisms and how they interact. He created work that bridges disciplines of biology, computer science, and robotics, and also that will have implications for regenerative medicine, cancer biology, and bioengineering. And yeah, he has multiple awards for these significant contributions in this field. So yeah, it's really an honor having you here. And yeah, I would say if that's okay with you, we dive right into your work.
Sure. Well, thank you. Yeah, a very kind introduction. Thanks.
Wonderful. So you published this paper in cellular signaling pathways, um, and this protocognitive systems, and how it will have implications for biomedicine. I thought it was really interesting to read, and especially you mentioned this idea of exploiting the collective intelligence of cells, and it kind of, in the recent papers you published, this comes kind of as a as a way of thinking or strategy, again and again, where you also then published the work about changing our view that cognition doesn't just happen in the brain. We should consider more systems. So would you like to maybe explain the audience a little bit about how you came to basically shift the paradigm in this way, based on your research? Thank you.
Sure, sure. Well, let's see. Let's start this way. There are a lot of people that are interested in this question of mind in the universe, and the question is, what kind of systems have it? Where is it found? How do we recognize it, and so on? And a lot of folks tend to discuss these things kind of on a philosophical basis. So they have they have certain beliefs that they're committed to, and so some people say that minds are only associated with brains, and then some other people see minds everywhere in nature, and other people really see it nowhere, and so on. And so there's a wide range of opinions, and people have been arguing about this stuff for a really long time. So to me, uh, what we need is not more philosophical arguments. I take an engineering perspective on these things, and so to me, all claims of intelligence, cognition, well, you name it, any of these kinds of terms, what they really mean to me is protocol claims, engineering protocol claims. So when you tell me that a certain system, and it might be a set of molecules, and it might be a thermostat, and it might be an autonomous vehicle, or a some kind of animal, or a synthetic a piece of synthetic biology, or some sort of AI, or a human, or an alien, or pretty much anything. When you say this system has some particular level of intelligence, or problem solving capacity, or agency, or whatever, what I hear is a particular engineering protocol for what is the best way to interact with that system. So, for example, if you tell me that this has roughly the status of a mechanical clock, then what that says to me is I shouldn't spend any time trying to reward it, or punish it, or convince it of anything. The only way we're going to be able to change how it works is by physically rewiring it. So it's sort of the most the you can imagine a spectrum. I call it a spectrum of persuadability, and so you can imagine something way on the left of that spectrum. And then if you say to me, well, actually this object, this system has the ability to do a kind of homeostasis, or it can do it has very simple goal directed action, then I'm thinking, well, then what I can do is I can find where the set point is stored, like in your thermostat. You don't really have to know how the whole thing works. You don't have to rewire it when you want it to go to a different temperature. You just need to know how to read and write the set point. Or you might say, well, this system is even better. It actually can learn from experience, and it can do maybe, let's say, Pavlovian conditioning. And I think, then that's even better. Then I really don't have to try to micromanage it from and control all the pieces. What I do need to do is provide some kind of reward and punishment, and various experiences to the system, and it will do certain things. And then you might say, well, no, this is an even more sophisticated system, and it actually responds to rational arguments, and that's even easier. I really don't have to do much at all except provide a rational argument. The system does the rest. And these are just sort of examples along the way on that continuum. So to me, all of these kinds of debates about intelligence and all of that boil down to some kind of hypothesis about how can you use this thing in an engineering fashion. In other words, what can you expect it to do when you're not there to micromanage it? And then we find out if that hypothesis is true or false. So all of these things are not up for philosophical debate. They have to be settled by experiment. So when somebody says this definitely can't be, they look at a particular thing, and I'll give you an example momentarily, but they look at something, they say, well, that can't be, it can't have decision making, it can't have memory, whatever. I don't think you can decide any of those things from sitting in an armchair. I think you have to do experiments, and when you do experiments, you're often surprised. And so in this paper, what Juanita Matthews and I and two undergrads students decided to talk about is this issue of pathways in the body. So this is molecular pathways that control health and disease and everything else. So these chemicals that interact with each other, and of course also not just chemical but also electrical and biomechanical and so on. But the question is, where on that spectrum does this sit? I mean, the modern assumption with molecular medicine is that they sit sort of all the way to the left, that they're these mechanical kind of mechanical types of systems, and even though they might be complex, there's really nothing there other than kind of an open loop emergent complexity. And if you're going to change how they act, you have to rewire them. And so we're talking about gene therapy and protein engineering and things like that. But that's a hypothesis. I mean, people assume that that's what it is, but we don't actually know that. And if you think about it, just boiling it down to two very simple examples. If you have a bowling ball sitting on top of a hill, then you know what you have to do in order to control where that ball goes. You really don't have a lot of options except for physically intervening with that landscape. You have to change the landscape in order to change where the ball is going to go. There's nothing else. There are no targets other than that. But if you've got a mouse sitting at the top of a hill, it's a completely different story. Using the equations you used for a bowling ball is not going to do the trick, because the mouse has beliefs and preferences and memories and other things related to how it sees that landscape, and it may be completely different than how you see the landscape. And so what you might do is actually give it stimuli to change how it perceives the landscape and the various payoffs of going in different directions, which is not something you could do the bowling ball. So that raises an important question. The body's pathways that we want to target for biomedicine, are they more like the bowling ball, which is what everybody assumes, or are they more like the mouse, or like something in between, or like something else? And so we talk about all the different examples throughout medicine and biology where it's pretty clear that the structure of the body has these amazing features that are absolutely more interesting than simple open loop mechanical systems, and thus that opens up the possibility for regenerative medicine addressing injury and cancer and birth defects and so on, to actually manipulate that kind of information processing and problem solving. The body has all kinds of competencies that we can take advantage of. So that's kind of the background to that paper.
Yeah, it's really interesting, because I thought a lot of times about this placebo effect and why
Mm,
can we use it to our advantage and kind of trick the system um, more systematically. So and you mentioned that also in the paper, more specifically. Do you have like a specific strategy, starting point to, to very practically, how could we leverage that?
yeah.
And do you have thoughts about that?
Sure, sure. Yeah, there are many. I mean, we've done some of it. We've already done, and we've been doing it for years without explicitly saying so, because it takes some time to be able to kind of put these things in the mainstream. But I'll give you a very simple example. Let's think about a gene regulatory network. So this is basically just a framework in which you've got a number of genes. So let's say, I don't know, a dozen. It might be more. It might be less. It doesn't matter. So just some number of genes, and they all turn each other on and off. So some gene might turn on a bunch of other genes at a particular rate and might turn off other genes. So you can draw this, and you have this complex network of genes turning each other on and off. And these things, these gene regulatory networks, they control embryonic development and physiology and all kinds of processes. Very, very important. And so you might look at that and you might say, okay, so what are my strategies for controlling how this thing works? For example, to improve a state, or to resolve a disease condition, and so on. And so one idea, of course, is gene therapy. So you might want to put in new genes, or delete some of the nodes, or change the way the nodes interact to try to control it. Or you might make the hypothesis that what if this thing is smarter than it looks, and what if it can actually change its behavior as a function of experience? And so we have two papers. One just came out this year, a couple of months ago, where we took 60 or so known models for gene regulatory networks. That people have studied in real cells and real organisms. And what we basically did was we said, okay, let's pretend that each one of these things is like an animal. It has some kind of learning capacity, and let's just ask, what types of learning can it do? And what I mean by that is, let's just imagine, let's the simplest thing. Let's think back to Pavlov's experiments, right? So the kind of classic Pavlovian conditioning. So you might, in that spirit, you might have a dog as your subject, and you might ring a bell, and the dog doesn't care about the bell. But after a while, if you pair the bell with the presentation of some food, so the bell in that case is your conditioned stimulus. You've got the food, which is an unconditioned stimulus, because you don't need to do anything for the dog to salivate when it smells the food. And so you produce those two. You pair those stimuli together a number of times while the dog learns, and after a while, what you can do is just ring the bell, and the dog starts salivating, because that connection has already been made in this system. So you can do this with a gene regulatory network. So let's imagine that you have some drug that that works well at the bench in a petri dish. It works really well, but you can't give it to patients for whatever reason. Maybe it's too strong or something. And then you have some other drug that normally doesn't do anything, and you have some kind of response that you're interested in. So you have three things. You have the node that you can stimulate with a dangerous drug, and it turns on the response you want, but you can't really keep giving that to patients. They won't tolerate it. And you have a kind of a neutral drug that doesn't do anything. Well, what our data show is that for the vast majority of these networks, if you just present together the two stimuli, so you tweak the node that responds to the strong drug, you tweak the node that responds to the neutral drug, and you just do that together. You just present them together some number of times. Eventually, what will happen is that the neutral drug alone is going to cause the response you're looking for. Now, in effect, that is a placebo. That basically it's a kind of molecular placebo, because you're causing an effect by using a drug that doesn't actually cause that effect. Except now it does, because the system has learned to believe. Basically, it's a simple version of associative conditioning that the presence of that drug really indicates that particular response needs to be turned on. It's the same thing as when people who are given a sugar pill by somebody in a white lab coat. They believe that they're going to get better because they have that association between those two things in their mind. And so that in effect is a kind of molecular placebo right there. And so one way you might use this is to realize that it's not a matter of forcing the network to do particular things by giving it a constant flow of particular drugs. It's the experience that network has via being exposed to different patterns of drugs according to different kinds of training paradigms that can radically change how the thing acts in the future. It's a learning system. And we had an amazing talk in our lab by this guy Fabrizio Benedetti, and his work. If you haven't already seen it, you should take a look. It's amazing. And he has a quote on one of his initial slides that says, "Words and drugs have the same mechanism of action." And I think that's really profound, because what our bodies do is use an interface, and it's largely a bioelectrical interface, but they use an interface between high level cognitive beliefs and goals, and transduce that down to the molecular action of your cells. Now, it sounds kind of weird. It sounds like we're talking about these very esoteric, sort of yoga and mind body medicine and meditation and placebo effects and voodoo curses and things like this. And it's actually not that at all. It's what your body is doing 24/7. So when you wake up in the morning, and you have this high level executive goal that you realize that you better go to work, in order for that to happen, that high level cognitive intent needs to change the molecular state of your muscle cells. It actually needs to depolarize your muscle cells to enable your body to get up out of bed. And so all the time, this is not some weird rare kind of mind body trick. It's how all creatures are made, that there's this transduction from high level informational states and goal states and beliefs and preferences and things like that down to the molecular events in your cells. They literally drive changes of molecular events. That's how we behave all day long. And so it becomes less bizarre that your beliefs can control other aspects of pathway function. If your thoughts and your plans can control the function of your muscle cells, why can't it control other molecular events in other cells? And so that's what people like Fabrizio study.
Yeah, it's really fascinating that, this kind of conditioning works on so many different levels because, I did a lot of, field conditioning and so on, the PhD
Hmm.
students in the lab and later. Um, and we looked also at molecular processes that these kind of elicit. So you just made these connections that people didn't make before. So it's really interesting to read that because once somebody made the connection, it is so logical. But
Yeah.
before that, you have to kind of get there,
Yeah.
and um, and that is what is so fascinating about the work and these recent contributions to the field. And so Do you think for future preventive medicine, do you think we can basically make predictions of what people will be exposed to or go through, and do this kind of condition training early on? You know, vaccine is one type, but like a larger scale vaccine, basically, and maybe a personalized one that protects maybe people from different mental health disorders and to cancers and infectious diseases that we might predict to come up by doing this training. Do you think by doing this kind of training early on in life, this gets conserved and we can elicit that memory fairly easily to kind of build up resilience preventively?
Hmm. Well, that's a really interesting hypothesis. I don't know. We don't know how long it'll stay. When you do this, it may be something that you need to redo from time to time. It may be something that lasts a really long time. Um, it may be that. Yeah, I don't know. This is just the beginning of these things, right? So a lot is still unknown, but we're certainly going to find out. This is exactly the kind of thing we need to know about.
Yeah, it's really interesting if we could, let's say, prevent PTSD in soldiers, or
Yeah.
you know, by pre-training that way. So, something like you give a drug that you know that if you know early on, some drugs can prevent PTSD, just people don't take it. If you could just take it when you're in soldier training,
Mm,
and then it will extend. That would be quite yeah, quite helpful.
yeah.
Um,
I mean, they
and
do have, like preconditioning protocols, right, for cardiac and for other types of organs. They have this kind of like for brain. I think that exists. This sort of preconditioning, how long it lasts, and what the ultimate uses are going to be. It's very hard to tell right now.
yeah, yeah, I did a little bit of work into PTSD and analyzing with machine learning. It goes a little bit away, but just, it was interesting because you have way more higher risk of developing PTSD if you're a firefighter than if you're a soldier.
Mm,
And we went to training through training paradigms, and because we found in language that using we way more often is kind of protective instead of using just I all the time.
mm,
Kind of. But anyways, and the soldier training is goes a lot into not seeing yourself as a separate item, to
mm,
see yourself in kind of a um, organism, kind of.
mm, mm, yeah.
And this was very preventive. So yeah, I know people are already using that, but maybe using it even, with additional drugs and so on would be really helpful. And yeah, you started earlier talking a little bit about the new model of cognition that you presented in the Frontiers paper that you wrote with two colleagues. Um, this was really fascinating. I thought about this before too. So, what do you think? This embodied cognition and distributed cognition, basically, to viewing it that way, how do you think it will change, like, how we view mental health disorders, and then also how we solve when a problem comes up? What do you think, or for the future, you would like to see happening based on that?
Yeah, I mean, to be clear, I'm not an expert on mental health, so take everything I say with a grain of salt. There, okay, I don't have any particular training in mental health medicine. Um, I think that what's really important about views like this is that they facilitate research by making it possible to take techniques from other fields and apply them here. So this is a powerful tool, and people sometimes say to me, "Why, you'd have less arguments if you just invented new words for everything. Like, don't use the same terminology as they use in cognitive science for memory and learning and goals and whatever. Just come up with new vocabulary, and then nobody will be upset." And the thing that you lose by doing that is you lose the opportunity for unification, for being able to test the idea that no, it really is the same thing. It's not some other weird kind of thing that just looks like memory, but it's actually something else. If you're willing to take the hypothesis that no, it is actually the same thing, then what you can do is you can borrow techniques from these other disciplines. And so we use in developmental biology all the time. We use tools that we take from cognitive and behavioral science, and they work really well. It's amazing. They can't tell the difference, and if they can't tell the difference, there's a reason we can use optogenetics and all the drugs and all the concepts like active inference and perceptual control theory and everything else. We can use and they don't distinguish between brains and other types of systems. And so that suggests to me that it's the distinctions that we make are largely arbitrary. We have these different departments and different journals and different funding bodies and different programs of education, and I think that's really problematic because it prevents you from borrowing these really powerful tools that already exist. And so I think using them in medicine is going to be huge. I think it's going to make a big difference to addressing developmental biology in terms of birth defects and inducing regeneration after injury and reprogramming cancer or the loss of multicellularity, which is basically what cancer is. What it's going to do for mental health, I don't exactly know because that's not really my area. But I think uh, really understanding how individuals arise out of components. So this sort of the grand mystery of life, which is you start off as a large collection of individual cells, and then eventually you become one embryo, one individual. So how did you become one out of 50,000 tiny cells? That that process is really critical, and I think if we understand that process, we're going to do a lot better in understanding how that happens in the mind and how what the various disorder states might be. And I think it's amazing and not often talked about that Alan Turing, who was very interested in intelligence and computation and different embodiments of mind and machines and all that, he also did a paper on chemical morphogenesis, the emergence of order in chemical morphogenetic systems. And people wonder why would somebody who was interested in all that stuff, why would he be looking at chemical morphogenesis? And I think my guess is that he saw pretty clearly that the self construction of the body is the same problem as the self construction of a mind. And it's really fundamentally the same set of questions. And if we understood one, we would be far further along to understand the other. And I think that will ultimately have a huge impact on how we treat various kinds of mental disorders, and in particular the big issues around dissociative kinds of dissociative identity kinds of disorders, and many other things. But again, I'm not an expert on that stuff.
Yeah, I agree, and I, since this very interesting paper came out, I don't know, it was 8 years ago, I think, where um, the lab that showed for the first time that there is an immune system in the brain, and then also that signaling from the immune system directly changed the activity of inhibitory neurons, and then directly change social behavior of mice with like immune deficiencies while they had an inflammation going on. And it was so direct, this change. When you think, as before, as a neuroscientist, social anxiety, you have these brain regions, these type of cells, and these transmitters, and so on going on. And then, the immune system comes in and surpasses all these things and just directly messes with that and changes behavior. That kind of, sorry, that kind of changed my view significantly, and that's why I was so happy to read the paper you recently wrote, because I think we can make so much more progress by merging these systems, especially the immune system, the neural
Mm.
system. And do you think that also, all the computational advancements we made, that these will now enable this to look at big data sets and make connections like these with achievements in AI, and how are you using them for this purpose?
Yeah, I mean, yeah, big data sets are important, and I think AI, of course, is going to be really really useful in analyzing some of these. But I think beyond that, we also need a lot of kind of conceptual work. So being able to look at the data is always sort of reflective of first being able to ask the right questions. And so, so some of the AI tools are sort of unsupervised and so on, and so that's good. You can learn things that you didn't know were in the data, but for a lot of it, we still don't even know how to ask the right questions. I think we're in many ways we're still groping for the right framework. So I think these big data sets are one part of the puzzle, but actually some of the conceptual work to really understand what's a good framework with which to look at the experiments that we're doing and what experiments we should do next and so on. I think that's different, that's distinct and sort of complementary to getting big data. That's a whole separate thing.
Yeah, I agree that the asking the right questions is the most important one. And a more how did your system, you as an individual, how do you think, what were the ingredients that you tend to ask and these right questions, and also that you make these connections that not everyone else is doing? So what do you think was important or is important for you to do that? Could you give advice for other people to also maybe make these connections?
Yeah, I can try. It's really hard to give this kind of this is sort of large scale meta advice because everybody's mind and goals are different. But I mean, for me personally, there were a couple of ingredients. One is that I didn't have a standard education in biology. I was a kind of a software engineer first, and did computer science, and then sort of biology. So I already was thinking about things in an unusual way. I was interested in this stuff from a very young age. As a kid, I was interested in both engineering and machines of various types and electronics and computers and so on, and at the same time the living world outside, bugs and insects and things like that, and what might be the difference between them. And uh, and and I like to read very widely. So uh, over the years, when I had time to kind of read more books and things like that, it was a lot of very different things. So it was a lot of philosophy and especially philosophy of mind and psychology and some mathematics and some other stuff, in addition to straight up engineering and physics and computer science and AI and things like that. So thinking broadly, I think is helped by learning in these other fields. I think a lot of people go through school and they might do very well and be very successful, but then there's not really time. They're sort of hyper specialized and really pushed to focus on very particular things, and that there's really not a lot of time to kind of go broad. So that's something I kind of recommend. And uh, I also recommend thinking about things from um, kind of a very different perspective, in the sense that, like, if I give a talk, I always know what kind of department I'm in based on which thing makes people mad, because some of the things I say are completely obvious in one department and are considered total heresy in another department. And so that tells you right there that there's a real set of differences in how people think about things. And so it's good to put yourself in somebody else's kind of position, and ask, okay, what would this type of person think of this from that vantage point, from that field? What does it look like from different types of perspectives? Uh, yeah, things like that. And it's also, it's also good to think about what kind of think about the end game of different fields. Like what would a when you're trying to answer a particular question, you might ask, what would a satisfying answer look like? So what would I even recognize a good answer? What would a good answer to this look like? So um, you might think, for example, if somebody's, I'm just making just as an example. I mean, I don't particularly study consciousness at all, but just as an example, if you're interested in a theory of consciousness, you might ask yourself, what would a correct theory of consciousness output? What format would it give predictions in? What does that look like? Or in regenerative medicine, you might ask, if we were to solve this problem, what does a solution actually look like? What is that? The final kind of the ultimate theory that allows you to resolve this. What would it have to do? What would it look like? And things like that. And that really helps to work backwards, because sometimes when you do that, you realize that actually we may not be asking the right question, because we don't know what a good solution would look like. It's hard for us to say what form it would take or what it would actually do, and that helps you to rethink the whole thing, and thinking in kind of counterfactuals is also very useful. I think it was Feynman that said once that people used to think that the Earth that the sun goes around the Earth because that's what it looked like. But he said, if they had stopped to ask, well, what would it look like if it was the other way around, they'd realize that, yeah, it would look exactly the same way. And so that would then lead you to the hypothesis that, well, then maybe who knows, maybe it is the other way around. And so asking asking yourself, what would it look like if if various things you believed were wrong? What would the what would that look like then? And for the things that you believe, or for the things that other people believe, these kind of counterfactuals, what would have to be true if we were completely mistaken about various things? So that's all helpful too. So those kinds of ways of thinking are things I find useful.
Yeah, thank you for giving us that insight. I think there are really important points in there. Um, so yeah, I think um, that I think this kind of question sometimes, people that are just starting off, it's good to have like guidelines like this based on what their decision is, what they would like to do. I think we need probably both, like specialists, and then people that also have kind of a broad overview. And you know, eh, when when we invite, for example, a guest speaker that mostly focuses on neurons, or and then we have here a few people, especially Serena. She likes the idea of how important also glia are. It's not easy sometimes to get people to think about, just two systems that are still part of the neural um, network, and to include them. I mean, there's a shift, but to practically get people to then include even more levels of complexity. And then do you does NIH come to you and ask you for advice how to shape grants? Because that would be important, right? To give like incentives so that the actual lab research goes more into adding levels of complexity. So what do you think?
Yeah, well, that's a really hard question. So I've certainly spent had my share of experiences on review panels and designing and implementing various funding programs. It's really hard to do this correctly, because we don't really know. You might ask yourself. what percent what's the ideal percentage of wild ideas? So if you're in charge of the science research budget for some particular country, and you're going to get thousands of applications, some of them are very pedestrian and kind of traditional and incremental, and then others are really wild moonshots. The question is, what percentage of the wild ones do you want? Because if you have a high percentage, most of those don't actually pan out. It's gonna waste the limited funds. But then if you have too few, then you're not going to make good progress, because you'll always be just doing incremental kinds of advances. So what's the right percentage? I have no idea what the right percentage is. I personally, of course, prefer the kind of the wild ideas myself, but that's not something that is necessarily sustainable at large scale, because most of those actually don't pan out to anything. So I don't know what the right number is. Now, one thing about the funding landscape is that there are lots of programs, even among the traditional funders like NIH and NSF. There are lots of programs that are kind of they're called these unconventional wild ideas. Supposedly you don't need preliminary data, you just need a good idea and all of that. So the problem with those, the program officers that design these are very well intentioned. They are literally trying to bring in new ways of thinking and give a chance to kind of new more interesting ideas. But the problem is, once you get those applications, somebody has to review them, and then they get handed out to the scientific community, and the vast majority of the scientific community, as obviously is the case, are somewhere in the middle of this curve. They generally tend to be pretty kind of risk averse, and very critical of new ideas. And even the ones who are pretty adventurous in their own narrow area are pretty risk averse and traditional in every other kind of area. And so these traditional reviewers generally do not review kind of any kind of unusual or iconoclastic kind of thinking. It's just they just don't do well in review. And so that's a real problem, is that no matter how much you try to encourage these kinds of applications, in the end they get reviewed by more or less the standard community. And of course the standard community views things in standard ways, and so they don't end up taking a lot of chances with things like this. And I don't know what the right percentage is. I mean, I personally wish we had more of that, but I don't know what the optimum number is.
Yeah, Joyce, please go ahead, and Kirko, please go ahead with your questions. Thank you.
Okay, I was going to say, unfortunately, I missed a lot of this, but I'll say I really do like the idea of increasing complexity of how you analyze things, and I'm going to look at your paper. And I also like the idea of bringing insights from other fields, because that's sort of my situation. But anyway, I also wanted to mention about that problem with grants and all. And I found out in detail about this when I went to a workshop held by the National Institute on Aging that I
Mm,
heard about, that was online. And I found out from this workshop that it was all on the potential infectious etiologies of Alzheimer's disease.
mm,
And all this research came out of the fact that they had found a big problem, just like you were describing, that they were not getting the funding, the grants going to people who wanted to study infectious etiology of Alzheimer's. And so they had to create a sort of a new parallel program, and I guess maybe have a different set of reviewers, because otherwise all the traditional view Alzheimer's researchers would deny,
yeah,
and low
yeah,
rate the grant. So anyway,
yeah,
that came out a really wonderful online workshop, and a lot of people are making progress in this area now that they figured out how to get around that problem.
yeah.
Anyway, thanks.
Yeah, thank you. No, that's a great example. I mean, yeah, it is hard, because what happens, and I've been on panels like this. What often happens, I mean, I think your community is very fortunate that it found enough people into that particular hypothesis to even have a workshop and find reviewers. That's great. What I've seen happen is somebody will do a program like this where they really try for unconventional, novel ideas in whatever field, and you find people that are interested in unconventional, novel ideas, but each of them has their own unconventional novel idea, and what they don't like is anybody else's unconventional novel idea. And so you have this review where it's just there's often just not enough people, especially at early stages. I mean, eventually things catch on, and then it's sort of you've got a bigger pool to choose from. But especially early on, there just aren't enough people that are able to competently review. And let's face it, this is a really hard task. I mean, NIH currently funds at something like 8%. Now, does anybody really think that we can distinguish an 8% quality grant from, let's say, a 12% quality grant? Because that's the dividing line is right there. And especially for novel ideas, we can't tell the difference. We can tell the terrible ideas from the pretty good ideas, but we can't. It's impossible, especially if it's not exactly our field, and often even if it is our field, to tell the genius ideas from the sort of wild but almost certainly not going to work ideas. It's just very hard to tell. So it's a the people on these study sections really do their best. They try really hard. They're told to discard 92% of the applications that they receive because they can only fund 8%. And now they're doing their best to try to figure out which of these things are likely to pay off. And it's a very hard task.
Yes, yes, thanks for your comment. I can see that. Yeah, and I also wonder about the whole area now of the microbiome, and there's a lot of research coming out where they're finding a microbiome in healthy patients' tissues, all sorts of their tissues, and it's kind of having a hard time, really convincing everyone that it's not, like error.
Yeah, yeah,
I'm convinced by the data, but not everyone is, and there's
yeah,
kind of a battle going on.
yeah,
But it makes another added level of complexity when you're not just looking at your own cells, but other cells from other microbes, trying to mess you up
yeah,
or
yeah,
doing something anyway.
yeah.
Yeah, hi Kyle. Did you want to ask a question or comment? Thank you.
Hi. Yeah. It's nice to meet everybody that I haven't met. I guess that's Kirko and Michael. Last night, Michael, we reviewed a paper. Katerina reviewed a paper, and I was present about a lab grown meat and the carbon cost of producing this, and it got me interested to be here today and talk with you. And just based on what you guys have been talking about, I had to ask Chat GPT. I have this a thread that's probably about 350 pages of a specific thread that's related to a model for uh, essentially imbuing what I call scalar conscionability, but it's effectively a consciousness model um, into artificial intelligence. And there are some key concepts that I brought into it that uh, I can go into the theory, but I thought it would be interesting to ask it a specific question. First, I asked it, um, could could you provide a synopsis of novel utilities discussed in this thread related to computational biology and chemical morphogenesis, in terms of how such utilities could advance our understanding of key concepts such as cellular signaling pathways? And it gave me a great little write-up that I could review as far as the theory behind this. So then I asked it, interestingly enough, could you hypothesize a novel discovery that could be made in computational biology, such as how to reverse cancer growth with lossless precision? One of the key concepts is this idea of what I define as a paranormal boundary, and there are very closely related concepts already out there, such as in string theory, there's the notion of a D brain, where open strings can end. This is sort of an open bilinear D brain, and it allows for um, complex systems with divergence, like scalar divergences, to be accounted for. So what it says, it started off as saying, firstly, reversing cancer growth with lossless precision would require imaginative thinking and exploration of potential avenues, which is currently an open question in computer science. Is can you define a phenomenal state? Can you define an experience, which I define as a discrete temporal derivative in this model? And it went on to say that it could hypothesize how this particular model could be used. I'll read here. Through extensive training and optimization, this computational model would be capable of accurately predicting the specific genetic and cellular alterations driving cancer growth in individual patients. By identifying the critical signaling pathways involved, this model could then propose precise interventions to reverse or inhibit cancer growth. The model's predictions would be validated through experimental studies where novel therapeutic approaches targeting the identified pathways are tested in preclinical and clinical settings. The goal would be to develop targeted therapies that directly interfere with aberrant signaling processes driving cancer growth while minimizing off-target effects. And it goes on to say that, that it could be continually updated and refined using feedback from experimental results. But this model effectively has an epsilon and sensitive loss capacity where it can divide space-time into discrete supersymmetrical folds and can deal with basically what I refer to as like digital drag. It's like quantum gravity effectively, but it does this by dividing space-time into these discrete variable boundaries, and then it can act on those.
Yeah, I mean, that all sounds interesting. I'm not sure. I don't know what I can say. It sounds interesting. For these kinds of things, in the end, the trick is always going to be what's the actionable outcome. So what's the experiment? What's the testable therapeutic? Like whatever the approach is going to be. That's often the tough part about these things is to go from the conceptual theories to an actual practical intervention that can be attempted in the lab.
Yeah, it got quite stumped that after it said here is one speculative hypothesis, it sat here for about 5-6 minutes before it spit something out, and it just kind of reminded me that the algorithmic framework is one thing, but developing proper hardware to run these systems and to actually utilize them would be another thing. And it's this particular, and you alluded to it, and it's I recognize this, and it'll be an interesting story one day if this thing's pan out well, and I look back on sort of decisions I've made involved in getting to this point. But it is this like black box problem that the ability for a particular model like this to reach its potential. My perception right now is that it's like multiple fields in tandem are depending on a spatiological model or metrological model to make improvements. And there was another talk that Katerina hosted with Lam Research, who does microetching for semiconductors, and they are limited by the same problem. So it's interesting, and I agree that it's the application. It's like this all these. It's a bottleneck of information distribution right now for me in this topic. So.
Yeah.
Yeah, that actually was a question I was about to ask is, these sort of models that you present for like organic organisms, do you think the same principles for making innovations in robotics, or maybe combining robotics with organoids, will the same will these principles also improve robotics in the future to have a more integrated systems?
Yeah, I think so, and I've written some stuff about which aspects of the biology can be used to improve functionality in engineered systems, both hybrid systems such as hybrids and kind of functional joinings of computer technology and living tissue, and also just for straight up engineering and robots and robotic swarms and things like that. So for sure, there are aspects of this having to do with multi-scale competency and specific things that cells do to make decisions about where they end and the outside world begins, which is actually a really important thing that living things have to figure out from scratch every single time, whereas our typical robotics have that determined from the beginning by the engineer, which then limits the system in important ways. So there's tons of stuff like that. I will say that actually I've gotten much more circumspect about writing those things, and I had started on a paper about some of that, and I don't think I'm gonna continue with it, because my concern is that the closer we get to a proper understanding of what are the features that give living things their special worth in terms of moral consideration. So what makes an animal worthy of various kinds of protections and so on. We're getting closer to figuring that out, and if we start imbuing technologies with those kind of considerations, that is very easy to copy. So it's very easy to make trillions of copies of something in a software kind of scenario, or even in engineering. It just creates it has the potential to create massive amounts of new entities that we have to worry about in an ethical sense, and I don't myself want to be responsible for that. So I'm not talking about risks to us. I'm talking about creating systems that, right now I don't think we have any engineered systems that we have to be particularly ethically concerned about. But I do think that we're getting close to the point where we're going to be able to make some. It's not that far off, and that carries a ton of responsibility. So I think I'm not really going to add to that anymore. But I do think that that's coming, and there's certainly plenty of other people that are working on it. So probably doesn't matter what I do. But these kinds of fundamental principles that we learn from biology are definitely going to be potentially implemented in other kinds of technologies, or even in software. And and then they'll have much better robustness and better performance and better intelligence, and all of those things will come. But depending, if we do certain things, we are also going to have to worry about them the way that we are supposed to be worried about living creatures. So that carries a whole other level of responsibility.
Yeah, thank you for that insight, and I agree that it carries a lot of responsibility, uh, uh, but the holy grail, I guess, for engineers is to develop something that can rebuild itself and create offspring, basically. Like, a robot that builds its perfect replica or even improves it is probably the holy grail in, your work, that kind of harnesses the cell's intelligence, uh, basically. If we could translate that into robotics, that
Well, I
we
think
can
we can. Yeah, I mean, I think we can. I absolutely think we can. I think it's totally doable. We are learning now more about what it is that actually makes that difference. There are a few people working in the field that I think are onto the right thing. Then there's many people that I don't think are onto the right thing, but there's a few that are, and there will be more and more of that, and we'll figure it out. And I do think I don't think there's anything unique about and privileged about evolution. I think we can engineer anything that evolution can find with its kind of random search. I don't think there's anything unique about cytoplasm and the kinds of specific components that are used on this that just happen to be used on this planet. There are many different potential embodiments of mind, and there's no reason why, once we really understand what the critical components are, that we couldn't engineer those directly in other substrates and in hybrid substrates and so on. So I do think it's possible. I apologize. I've got about a minute before I have to go. I've got a meeting at 4.
Yeah, I was I was about to say that we are up to the hour. And is there any last comments, maybe some peek into the near future that you would like us to learn about?
The only thing I'll say, and I'll just leave it with this simple cryptic statement, and eventually I'll write something about this. But I think that as people think about AI and a lot of the concerns that people have about AI, I think we need to realize that those concerns, those potential issues, are already here with us in the biological world, and they're just much more obvious now. But people who really think deeply about these things have been wrestling with these issues for a really long time, and so what the AI is really doing is holding up a mirror for the average person now, not just for philosophers of biology and scientists, holding up a mirror to the issues that we as biological organisms have already had to deal with. And this is, I could list dozens of these things, but they're not unique to AI. They're really fundamental problems that are with us, and existential problems that have been with us and bothering thinkers in this field for centuries before we actually had AI. So I'll just leave it at that.
Yeah, well, thank you so much for taking the time. This was really a wonderful conversation that went through a lot of fields and different thought processes. So it's always a pleasure having you here. And yeah, I hope to read more about your work in the future. And I think all of us, and enjoy the rest of your day. And yeah, thank you so much. It was an honor. Thank you.
Cool. Thank you so much. And thank you for pulling this together. And thanks a lot, everybody. Be well.
Thanks so much.
Thank you. Bye.
Yeah, thank you. And thanks everyone for coming, asking questions, sending questions. And yeah, if you like discussions like this, join us again. We have a few rooms coming up. So yeah, enjoy the rest of your weekend, and yeah, come back and send in questions if you have follow-up questions or suggestions. Okay, thank you. I'll close the room in 3,
Thank you.
2, 1. Thank you. Bye, everyone.